Hey A&Pers, this is Dr. Jones checking in. This is A&P 1, Lecture Test 1. We're on Chapter 2, video number 11. So this is our hopefully last chemistry chapter. Yay! Time to be done with chemistry and move on to cells. All right, so the top of page 17, which for me looks like this. Let's dig in. Monomers of a nucleic acid are called, oh, we're in blue, nucleotides. Nucleotides have three parts. You have the pentose sugar, and the pent means 5, so this is your 5-sided sugar. And there are two different 5-sided sugars, pentose sugars, that are used in nucleotides. One is ribose in ribonucleic acid, also known as RNA, and one is deoxyribose, so the same but minus an oxygen, D without oxygen, deoxyribose, and that's DNA. So that's why the name difference between RNA and DNA. It's a different sugar, so it's named for the sugar that's different. Okay. Either ribose in RNA or deoxyribose in DNA, and then there's a phosphate group and a nitrogenous base. And there are 5 different nitrogenous bases that are found in nucleotides: adenine, guanine, cytosine, thymine, and uracil. And those are abbreviated with their first letter capitalized. I have a chart. Boom, chakalaka-laka. All right, so DNA and RNA both use adenine, guanine, and cytosine, but they do not use the same on this fourth one. This fourth column is different. DNA only uses thymine, and RNA only uses uracil. Okay, you never use thymine in RNA, and you never use uracil in DNA. So, unity of life, it's the same everywhere. And as we mentioned earlier, there were 3 types of RNA, and it's time to learn them. And these are abbreviated with the first letter of the word that describes them in lowercase, and then RNA in all caps. So mRNA is lowercase m, capital R, capital N, capital A, and this is messenger RNA. tRNA, T for transfer RNA. And then ribosomal RNA, which is rRNA, but the first R has to be lowercase, the second R and the N and the A are all caps. And this is ribosomal RNA. And we will come back to this in Chapter 3, which is still on Test 1, and talk about exactly what each of these RNAs do. And we said already that the function of RNA in general is to make proteins, and these will all work together to help synthesize proteins. T-E-I-N. I'm writing with a mouse, I apologize. Okay, so mRNA is the first one that we're going to deal with. mRNA is a single strand, while DNA makes a twisty ladder shape called a double helix. I mentioned this in the previous video. Here it is spelled out for you so that you can get it written down. The rungs of the ladder, these bits, are actually made of complementary base pairs. And I do want to point out, because y'all are college educated now, that complementary here is complementary with an E, which means they go together. Complementary with an I is, ooh, I really like that shirt. Complementary with an E means they complement each other, not, oh, you're so great, but like they work well together. Like white chocolate and raspberries, complementary. Oh, now I want white chocolate raspberry something. Okay, so the rungs of the ladder are made of complementary base pairs, and we went from that to chocolate. That sounds fair. And the complementary base pairs are a pair of 2 nitrogenous bases with weak hydrogen bonds holding them together. True or false? So, hint, anytime in the note set I could not think of a way to make the statement in the book a question, I simply throw a true false at the end of it. So in the note set, all the true false questions are true. On the tests, probably about 50-50, although I don't go back and edit them to make sure that they're exactly 50-50, so don't like count on them being 50-50, but it should be fairly random whether it's true or false on the test. The note set, it's true. That puppy is true, and I just couldn't think of a way to make it a question. So that's why. Okay. And in DNA, adenine binds only with thymine, and guanine binds only to cytosine. So this is our mnemonic from the Wood note set at the golf course. Remember that A and T go together, and G and C go together at the golf course. Because, as you see here, the adenine and thymine, they have 2 hydrogen bonds that hold them together. The cytosine and guanine are shaped differently and have 3 hydrogen bonds that hold them together. And so the adenine, the guanine, cytosine, the thymine, they literally cannot stick together. It's the wrong size zipper. Okay, like one side, and then nothing. Not gonna work. Okay, that was page 17, audible page flip noises. To remember these complementary base pairs, ATGC, think of the sentence at the golf course. In all RNA, of course, we don't use thymine. Instead of thymine, RNA uses uracil. That blue doesn't show very well on the lighter blue at the bottom of the page, so let's switch back to red because that'll show. Uracil. Okay, so uracil is used instead of thymine, so we've got AUGC. AU is the chemical symbol for gold, so gold golf course. Uber golf course. I have no mnemonic for this, so if you think of a great mnemonic for the RNA base pairs and how they complementary base pairs pair together, please leave it in the comments because I got nothing. Okay, help a teacher out. All right, now we have rules. So let's whiteboard screen. Actually, we're in red. Let's black screen. Ooh. Feels illicit. Illicit means illegal. It's not though. Okay, so we've got DNA rules. I'm writing with a mouse and I apologize and I know it's terrible. All right, there's the edge. I was just trying to find the edge. So adenine binds to at the, okay. Thymine binds to what? A, adenine. And guanine, we said golf course. The G and the C bind together. Remember they had 3 hydrogen bonds, whereas the AT share 2 hydrogen bonds. And then what binds to cytosine? Well, guanine. So these are the DNA rules at the golf course. So if you have an A and you need to find its complementary base pair, it's always going to be a T. If you have a T and you need to find its complementary base pair in DNA, it's always going to be an A. G complementary base pair is a C, and C complementary base pair is a G. All right, so now let's talk about RNA rules. All right, so we've got adenine, uracil, because remember there's no thymine in RNA, guanine, and cytosine. So in RNA, there's no uracil, so what goes here? There's no thymine, so what goes here? Uracil, you're right. Okay, so what's uracil bind to? A U golf course. The A, right? So these are bound together with the 2 hydrogen bonds. And guanine binds to cytosine. Cytosine binds to guanine. These are kind of getting to be old hat. All right, so now if on the test I say I have this DNA and we're going to bind it to DNA, what complementary base pair will bind to it. So these are the DNA to DNA rules. All right, in DNA, you have DNA, you're binding to DNA. What is the A attached to? That's right. And what's a thymine attached to? What's a guanine bind to? A C. What's a C bind to? A G. So these are the same. All right. Let's skip that one and go to, we're skipping one on the note set. We're going to RNA to RNA. So the messenger RNA binds to, well, the transfer RNA binds to messenger RNA. So if we're going from one RNA to another RNA, then we use the RNA rules. A, U, G, C. So if we have an adenine in RNA and we're making an RNA, what binds to it? If we have a uracil, what binds to it? What binds to guanine? The only thing that binds to guanine. What binds to cytosine? The only thing that binds to cytosine. So these are, sorry, drawing with a mouse, the same. The RNA to RNA rules use the RNA. The DNA to DNA rules use the DNA rules. Okay. Now we're going to change colors because this next part is different. Ooh, yellow. Ooh, I bet that'll be vivid. Okay, now we skipped the DNA to RNA rules, and these are very important, so we're going to make sure we've got these. So if I say on the test, and I absolutely will say on the test, you've got this DNA triplet, and we'll talk about that means when we get to chapter 3, when we get here, okay? And it's going to make an mRNA. And we will use the DNA to RNA rules. So these are different. Okay, well, let's start at the bottom and go up. What does a cytosine bind to? A guanine, only, ever. What's a guanine bind to? A cytosine, only, ever. What's a thymine bind to? Only, ever. Adenine. Here's the difference. What is an adenine in DNA bind to in RNA? No thymine in RNA. The adenine binds to the uracil. There's always some of these questions on the test. Always. But make sure you read the question carefully. Because if I say we're going from DNA to RNA, then you need to use the DNA to RNA rules. If I say, because the DNA actually replicates, so you'll use the DNA to make DNA, I say, okay, here's the DNA. What is the complementary base pair in DNA? Then you use the DNA to DNA rules. And if I say we're starting with mRNA and what tRNA bind to it, then you use these RNA to RNA rules. Okay, we will practice that. All right, let's finish this page and chemistry. This is a bit of review, but if there's any of this that I didn't say, definitely get this written down. The DNA shape is a double helix, while the RNA shape is a single strand. The DNA sugar is deoxyribose, the RNA sugar is ribose. The DNA, the different one is thymine of the nucleotides, and the RNA, the nucleotide that's different is uracil. Mnemonic here is at the golf course. Mnemonic here is gold golf course. AU golf course. At your golf course. I can't even do that. It's YOUR. It's not UR. Okay. And the function of DNA is to store genetic material. And the function of RNA is to make proteins, a process which I promise we will cover in great detail. And here's something I may not have said yet. The DNA at double the size stays in the nucleus because it's too big to leave through the nuclear pores. The RNA, however, is half the width and is smaller and does fit through the nuclear pores, and it goes out into the cytoplasm, which is a term we haven't learned yet, but we will. Give me like the next video, we'll cover that. Into the cell, out of the nucleus. At this point in the lecture, if you haven't gotten to the cell parts in lab, then you're behind in the lab material. Third blank means I'm serious, although that might have been 4, I kind of lost count. So my suggestion, and I hate to tell people what to do. I'm a rebel type, and if you tell me what to do, then that's kind of the last thing I'm going to do. So I'm all about giving people choices. However, as I have had lots of schooling experience, I get asked fairly often for recommendations. So if you would like my recommendation, here it is. If you would not like my recommendation, this is still my recommendation, whether you are interested or not. Study lecture and lab both every day. That's my recommendation. If you're not moving forward in both lecture and lab, especially in these online-only semesters, on a daily basis, then you are behind. There's too much material to learn all at once the week of the test. These first tests, there's more time for lecture and lab before the first set of tests than there are any of the other tests. All the other tests are like 2 weeks apart or 3 weeks apart. This one is put off. So if you're not studying every day, like the freedom to go at your own pace is just enough rope to hang yourself on. So don't. Be diligent and every day, every day, every day. So if you have an hour a day for lecture and lab, then that is recommended. And then once you've built that habit, then you work on twice a day. So again, not telling you what to do, but my suggestions of what the students who have done the best tell me they do, and how I got through school, and how my friends got through school, and all of that combined knowledge to try and help you if you are interested. So I do want to tell you the 24-hour rule, like say, because if we were in lecture and lab, you'd be in lecture for an hour and a half twice a week, so 3 hours of lecture a week. And same with lab, 3 hours of lab a week. So if you're not watching 3 hours of lecture videos and 3 hours of lab videos a week, you are behind. Okay, so 6 hours a week of watching the videos, reading filling in the note sets is minimum for just showing up to class. This is not the kind of class where you can just show up to class and then remember everything at the test, okay? That was high school, this is college. It's not like that now, unless you're way brighter than I am, which is entirely possible. We always get super bright students through these classes. So not saying it's not possible, but it's not likely. Most of us have to study. And so, if you don't know how to study, I will be making some study skills videos to help you with some of that. The first trick is getting yourself to sit your butt down and engage with the material. But you have to, on an online-only class, watch the videos on your own, and then also go back and review on your own. So say you sat in class for an hour and a half of lecture, an hour and a half of lab, a 3-hour block with me, and you understood everything I said in the entire 3-hour period. If you did not review that material within 24 hours, about roughly half of it would go poof roughly 24 hour by the 24-hour mark. And it's different for everyone, but that's kind of an average estimation. So your brain tries to protect you by ejecting material that's not important. So you have to review this material every day, every day, every day, every day. And once you've built that habit, then we'll work on twice a day. So if you, like, say, between a Thursday class and the next Tuesday, if you didn't review on Friday, then you've lost half the material. If you didn't review on Saturday, you lose another half of what's left. So now you're down to 25% of the material, assuming you understood everything the first time. If you didn't review on Sunday, then you're down to, what's half of 25%? 12.5%? Let's do fractions, half, quarter, an eighth of the material. And if you didn't review on Monday, then you're down to a 16th of the material. And if you don't review before class on Tuesday, then you're down to a 32nd of the material, assuming that you understood everything in the first place. So super important that you review the material after you learn it, because remembering it for the test is about getting it into not just your short-term memory, but start getting it into your long-term memory so that you'll be able to recognize the right answer when you see it on test day. And so to do that, you want to interact with the material in lots of different ways. That's why I recommend that you write things out as much as you can, or type them in if that's how you do. They do say that writing it out does make for higher quiz grades. So when they have done studies with quizzes, people who take handwritten notes tend to get a little higher. Again, it's a tendency. I've had lots of people who type and get straight A's, so, you know, it's just a tendency. Not a hard and fast rule. So, study. Lots of studies. Okay. All right, well, we will talk more about study skills later. Let's see, was that everything? It was not everything? Oh, I thought we were finished with this page. I was getting ready to wrap up the lecture. All right, let's finish this page. Sorry. A high energy bond is a covalent bond whose breakdown releases energy in a form the cell can use. One more time. A high energy bond is a covalent bond whose breakdown releases energy in a form the cell can use. There are lots of different forms of energy, like heat and light and microwaves and electromagnetic waves. Actually, everything I listed were electromagnetic waves. Oh, there's electromagnetic waves and gravitational waves, which you actually have proven exist, but we're moving on. So a high energy compound is a compound with a high energy bond. So the term phosphorylation means to add a phosphate group. Let me move me out of the way. Out of the way. Okay. So here's a phosphate group. Here's adenosine. Adenosine is an adenine with a ribose. So that together is adenosine. With a phosphate group added, it's adenosine. The yellow doesn't show. The yellow doesn't show. Okay, an adenosine monophosphate. For the mono for M, so that's one phosphate, monophosphate. So then when we phosphorylate it and add this, actually over here, add this second phosphate, then we create a high energy bond. And now we have two phosphates. So we have adenosine and di-phosphate. Di means two. And then when we phosphorylate again and add a third phosphate, we get adenosine triphosphate. Tri for three, like a tricycle has three wheels. So this is your vaunted ATP, which we'll be talking about for the next 2 semesters. Okay. And yeah. Adenosine and 1 phosphate, adenosine and 2 phosphates, adenosine for 3 phosphates for a MP, a DP, and a TP. And the chemical notation for a phosphate group is capital P, capital O, subscript for superscript 3 minus. That's so weird. Okay. And the synthesis of a high energy compound requires a phosphate, the enzymes, and the substrates, which are the other reactants, are substrates. The most abundant high energy compound in the body is ATP. The second one is GTP, which is the same except with a guanine instead of an adenine. So it's, yeah. All right. Next, we will start chapter 3. Dun, dun, dun. All right. Thank you, as always, for tuning in. This has been Dr. Jones with ANP1, Lecture Test 1, Video, Checks Notes 11. And we finished off page 17 and 18 and have completed chemistry. Yay! So we will come back with the next video on the top of page 19 with chapter 3, and we will talk about the cellular level of organization. Again, if you don't know the anatomy of the cell, go catch up in lab and then come back and watch the next video. The education must go on. Dr. J out.